Hello, everyone. This is Patrick Morrison from the Wolfpack Astrobiology podcast, and in today's video series, we're going to look at what evolutionary innovations preceded the Great Oxygenation event. The two major metabolic processes that have allowed life to have such a profound impact on the Earth are photosynthesis and respiration. Respiration uses redox chemistry as a means to harvest energy. Here, organic carbon, with the generic formula CH2O, is reacted with the oxidized form of another element, depicted here as A. The organic carbon is oxidized to carbon dioxide, while element A acts as the electron acceptor and is reduced. So long as the element acting as the electron acceptor is more electronegative than carbon, the reaction will produce energy which the organism can utilize. It does this by using the flow of high-energy electrons to pump hydrogen ions out of the cell. The inflow of these ions is then used to drive the ATP synthase enzyme, which regenerates ATP, the energy currency of the cell. If the reaction is reversed, an input of energy is needed, and organic carbon can be produced from carbon dioxide by taking electrons off the other element. In photosynthesis, this input of energy comes in the form of light. Today, oxygen is most often used as the electron acceptor in respiration, while the reduced form of it, water, is used as the electron donor in photosynthesis. However, other elements such as iron, nitrogen, manganese, and sulfur can be used as electron donors and acceptors, and here we'll discuss how these systems came into being. When in the history of Earth did these innovations take place? The second eon in Earth's history is the Archean, which can be thought of as the age of prokaryotes. The Archean began 4 billion years ago with the formation of the undisputed oldest rock, the Acasta gneiss, and ended 2.5 billion years ago with the Huronian glaciation event. The Archean world was very different from the Earth we're familiar with. The atmosphere was 75% nitrogen, 20% carbon dioxide, and 5% other gases such as water vapor, argon, sulfur dioxide, and methane. The oceans were salty, with most ions being sodium and chloride, but whereas sulfate is the third most abundant ion today, the oceans of the Archean were ferruginous, with high concentrations of iron 2 ions. Furthermore, while the oceans of today are alkaline, the Archean oceans were acidic with a pH of around 6. At the outset of the Archean, scattered islands were the only land that existed. The sun was between 75 and 85% as luminous as it is now, with greenhouse gases keeping the surface warm. The Archean is subdivided into the Eo, Paleo, Meso, and Neo-Archean eras, and in each one, it appears that a different element acted as the electron donor for the production of organic carbon. Because of this, each era can be seen as a different elemental age of metabolism. The Eoarchean can be thought of as the hydrogen age. Because hydrogen is less electronegative than carbon, molecular hydrogen can be reacted with carbon dioxide to produce both organic molecules for biomass and energy for metabolism. The Eoarchean spanned from 4 to 3.6 billion years ago, and the late heavy bombardment spanned this era along with the late Hadean. The mantle was some 200 degrees hotter than it is today, leading to elevated rates of volcanic activity. The crust and mantle had little time to chemically differentiate, so the crust itself would have been composed of mafic and ultramafic rocks. Both volcanic and serpentinizing vents were abundant on the seafloor. The oldest metasedimentary rocks, those between sedimentary and metamorphic, are dated from this time, with the oldest being found in Aquileia at 3.85 billion years old and Isua at 3.771 billion years old. Plate tectonics may have begun at the Hadean-Archean boundary, but this is disputed. Let's now take a look at the stable isotope record. Prior to 3.5 billion years ago, The only biophilic element which shows characteristics of biotic fractionation is carbon. Reduction of organic carbon by biotic processes will give biomass depleted in carbon-13, while abiotic processes will result in organic carbon with a similar quantity of carbon-13 as the source material. When the production of organic carbon by organisms is limited by the availability of carbon dioxide, the change in abundance of carbon-13 would be between what would be expected by biotic and abiotic processes. Diagenesis has turned the organic carbon from the Eoarchean into graphite, and the graphite from Achillea has an isotopic composition that's characteristic of abiotic and biotic carbon-limited processes. This is also true of most of the graphite found in Isua. Fischer-Tropsch type abiotic organic carbon synthesis reactions are common in serpentinizing systems, which are also carbon-limited. This, coupled with the isotope record, suggests that the Eoarchean biosphere was largely confined to these habitats. However, some of the graphite found in Isua has a carbon isotope composition characteristic of carbon-replete biotic production, indicating that life may have colonized volcanic vents, also known as black smokers, by this time. Many modern systems have their roots in the last universal common ancestor of all life alive today, or LUCA. This refers to a population of organisms which lived in a volcanic hydrothermal vent between 3.5 and 3.7 billion years ago. LUCA had a more primitive molecular biology than modern organisms, with DNA replication and transcription being carried out by ribozymes. However, the enzymes involved in its metabolism were comparable to those used by life today. LUCA was an autotrophic acetogen, meaning it reacted hydrogen with carbon dioxide to produce acetate. The flow of electrons from hydrogen to carbon was used to pump sodium ions and protons out of the cell to generate a cross-membrane pH and salinity gradient. This gradient was then used to power the ATP synthase enzyme. LUCA would have also taken up phosphorus as orthophosphate, sulfur as hydrogen sulfide, and nitrogen from either ammonia or hydrogen cyanide. It also had adaptations to cope with toxic chemicals emitted from the vent, such as sulfite and arsenite. What's important to keep in mind is that LUCA inhabited one of many environments on the Eoarchean Earth. The first major metabolic process which came into being after LUCA was likely methanogenesis. Acetogens use the Wood-Youngdahl pathway, which is comprised of a methyl and carbonyl branch, where electron carriers take electrons from hydrogen to reduce a pair of carbon dioxide molecules to acetate. Autotrophic methanogenesis omits the carbonyl branch and instead expands on the methyl branch to reduce the carbon dioxide all the way to methane. Once a carbon dioxide is reduced to a methyl group on an H4-MPT molecule, it can either be reacted with coenzyme A to be fed into biosynthesis pathways, or be bound to coenzyme M to produce energy. The resulting methyl coenzyme M is reacted with coenzyme B, where the methyl group is reduced to methane, and coenzyme B is oxidized, forming a disulfide bond between it and coenzyme M. Two electrons taken from a hydrogen molecule are then used by a heterodisulfide reductase enzyme to reduce the disulfide bond to form a pair of SH or thiol groups, regenerating coenzymes M and B. Acetogens have a single step where the flow of electrons is used to power ion pumping, but the addition of this last step on the methyl branch includes another transfer of electrons, which is used to pump ions out of the cell, increasing the energetic yield. Furthermore, methanogens possess electron carriers in the membrane, such as MPH2, which deliver electrons to heterodisulfide reductase, and porphyrins with an iron atom in the center, known as sterohemes, to facilitate the flow of electrons. In the Stem of Life series, we talked about how acetogens came about as a coming together of the carbon assimilation and energy metabolism, but methanogenesis can be seen as the initial decoupling of the two. Another process which is common across all branches of the tree of life that likely arose in the Eoarchean is fermentation. While redox chemistry uses the change in enthalpy of a reaction to produce energy, fermentation does so by increasing the entropy of the system. LUCA possessed enzymes involved in glycolysis and the pentose phosphate pathway, but lacked enzymes that produce volatile fatty acids such as lactate, which are often the end products of fermentation. Furthermore, the oxidative branch of the pentose phosphate pathway is not present in the abiotic analogs, suggesting that this was a novel process in biotic systems. Based on this, it's likely that the enzymes involved in glycolysis in the pentose phosphate pathways were used by LUCA to produce glucose and ribose, with ATP being used to power the reactions. Fermentation itself came about as a reversal of these pathways, breaking down complex biomolecules like sugars into simple organic acids to produce energy, which regenerates ATP. This process is known as substrate-level phosphorylation. One variety of methanogenesis, known as acetoclastic methanogenesis, is functionally similar to fermentation in that it breaks acetate down into carbon dioxide and methane, but it isn't considered fermentation in the technical sense because instead of using substrate-level phosphorylation, it uses the flow of electrons from the carboxyl group on the acetate to the methyl group to pump ions out of the cell. Acetoclastic methanogenesis feeds acetate where it would usually exit the Wood-Youngdahl pathway, where the carbonyl branch is reversed to produce carbon dioxide, and the downstream element of the methyl branch is used to produce methane.